Okay, so let's get started with the casual talk session together with selection committee members. So first of all, congratulations on your winning of the 25th Keio Medical Science Prize. So in this session, we'd like to speak with two laureates and about their research career and hopefully talk a bit about something we normally wouldn't get to hear about. Okay. So thank you for both for joining us today. So I heard, so it's the first time for both of you to meet this time. So I hope this opportunity will be great to, to start communication with you guys. So before starting for the audience, let me briefly introduce Dr. Regev first. So Dr. Regev graduated from Tel Aviv University in Israel, received a Ph.D. for computational biology in 2002, and became a core member of Broad Institute of MIT and Harvard in 2006 in Boston. And in 2015, Dr. Regev was promoted as a professor and chair of the faculty in Department of Biology of MIT and Broad. And from this year, became an executive vice president of Genentech Research and Early Development. And Dr. Regev has developed computational and experimental methods for single-cell genomics, namely how to formulate the core biological questions that single-cell genomics can address. So leading to the field ever since it was first introduced in the early 2010s. So Dr. Regev co-developed DropSeq, which enabled a large-scale single-cell analysis and further developed ProTabSeq to explore the impact of thousands of genes disruptions in combination with the CRISPR technology. So moreover, Dr. Regev has been playing a central role in developing the international community Human Cell Atlas. So single-cell genomics has opened up a new field that has yielded a broad range of fundamental discoveries into the function of cells and tissues in health and diseases, and helps to discover new therapeutics. Okay, so Dr. Regev, how do you feel receiving this prize?
Very grateful and very honored and very humbled, of course. I'm thrilled to receive it in such great company. And I'm also happy for the recognition that this has for both the field that I work in now, single-cell genomics, and also more generally for the impact of new technologies on the types of biological questions that we can ask.
Thank you. So next, let me introduce Dr. Miyawaki. So Dr. Miyawaki graduated from Keio University School of Medicine in 1987. Received Ph.D. in Osaka University in 1991, and spent a postdoc for 2.5 years, from 1995 to 7 in UC San Diego with Dr. Roger Chen. So in 1999, Dr. Miyawaki became a PI in RIKEN, and now is heading a cutting-edge lab in RIKEN Center for Brain Science and also Center for Advanced Photonics. So Dr. Miyawaki has developed many innovative fluorescent imaging probes that depict dynamic changes of small molecules in living cells. For example, during the cell cycle, Fuchi, and mitophagy, mito-SRAY. In addition, in order to detect signal from deep within organs, Dr. Miyawaki has developed SCALE, an optical clearing reagent for biological imaging, and Akabri, a new bioluminescence in vivo imaging system. So these new technologies are widely used by researchers around the world and has had a great impact on the development of the field of medical and life sciences. So Dr. Miyawaki, as a Keio graduate, how do you feel now?
Okay, as I said, I graduated from this Keio Med, so Keio University Medicine, and after graduation, I was rather away from medicine for a long time period. So now I like to contribute to medicine more than before. And well, so this prize giving us, you and me, to talk each other directly. And Well, I very quite interested in the combination, so your approach and mine to, for example, I like to propose a computational bioimaging because the introduction of functional probes into cells so may result in either upregulation or downregulation of downstream signaling pathways. And also, even if we adopt the knock-in method so for probe introduction, so I'm talking about fluorescence or bioluminescence imaging. Okay? So a substantial amount of light, chemicals, substrate should be absorbed by cells that accumulate so those functional probes. Okay? So seeing is perturbing. This is the point, right? And well, so the quantitative or computational bioimaging is expected to provide methodological framework for simulation, so with the observation-dependent perturbation. Okay? So many people say seeing is believing, but I do say seeing is disturbing or perturbing, right? So I think once we accept that notion, now so the process of the visualization can be viewed as a reaction, very interesting reaction toward the object. So I need your help, right? That's it.
I have several thoughts on that. First is, I loved what you said about seeing is perturbing, because to a large extent, that was the first way that we thought about single cell profiling. We thought about the variation between individual cells, which is just something we observe, not a perturbation that we ourselves introduced, as being basically a form of perturbation. Because in any individual cell, the variables are a little bit different from each other. And so we assumed that we could infer some of the, not just these, the types and the states and so on, but actually the circuitry inside those cells from these subtle variations across the cells. And we now know that that's actually, to some extent, at least quite possible. So that sense that the what we observe is actually a perturbation is not. I think it applies across a broad range of things. The other thing that I'm very passionate about, and I think that's where our two worlds actually finally can meet, because they're both at the right scale and resolution now that you can put them together, is this idea that at the end of the day, in the cell itself or in a tissue, it's not like there's image and there's molecules or genes. Everything that we look at is made from molecules. It's just that sometimes we don't know the mapping, meaning we can look at something and observe it under a microscope, and with a molecular probe, we could also know its molecular identity, but without it, what we see is structure. And we can take the same entity, say the cells, and we can profile them for their molecular constituents. But in reality, there's only one cell. The image that we see is actually made of molecules. We just don't know the mapping. And that is where I think the combination of the kinds of data we can measure today and the algorithms that are at our disposal can actually unify the molecular and the visual view of cell biology and make only one unified cell biology. And there's experiments that can make that possible. So for example, you can do today two types of experiments under genetic perturbations. You can do a perturb-seq experiment where you introduce a set of genetic perturbations and you get in return for each genetic perturbation the distribution of cellular states that go together with it through, say, single-cell RNA-seq or another single-cell genomics method. So you get for each perturbation a series of cells that carry the perturbation and the molecular profiles of these cells. But at the same time, you can do the same experiment using methods like pooled optical screens, where you introduce the same set of genetic perturbations, the most beautiful set of probes that one could imagine. You can monitor the cells, including through live cell imaging, through a period of time in the experiment. And at the end of the experiment, you can perform in situ sequencing and actually know for each of the cells that we have been imaging live, you can know the entities, the perturbations that were inside it, one or more. And again, you would get a distribution of cells with the images and the movies actually, and every beautiful thing that one can see with their eyes. And now this is where the algorithms really come in. So now you can take these two views of the world and here's a distribution of cells with molecular profiles under the perturbation. Here's a distribution of cells with images under the perturbation. Please match that to me. and not just do it for one perturbation at a time, but for a large series of perturbations. And what we get out of that is all sorts of wonderful things. For example, we can predict that if this is the molecular profile, this would be the image that goes along with it. But also, if you go to the model that makes those predictions and you dig in the insides of the model, you can say, oh, an imaging feature that looks like that goes with molecular features that are like so. And all of a sudden, you would get like a molecular painting back from the algorithm. And you can only imagine these things now because you can do these types of experiments in parallel. And 5 years ago, we couldn't, but now we can. And now that we can, and the algorithms are there as well, I think that's kind of the right time for this kind of unification. You can do it for cells, you can also do it for tissues.
I look forward to the algorithm for prediction. Yes, in many ways, yes. Thank you.
But for algorithms to predict, they need to be trained on something. And that's where the lab methods need to produce the kind of data that you can train on.
Okay.
Yeah, that's so exciting. So yeah, that's going to be a great marriage. Those two, cutting edge technologies get together and to get a new biology. Yeah. So Dr. Siomi, chair of the selection committee. So.
Hi, I'm very pleased. This year we have selected Dr. Regev and Dr. Miyawaki as the Keio Medical Science Prize winners, recognizing your creativity and ingenuity in developing a number of single-cell-based analysis techniques, which have contributed a lot greatly to basic biology and also medical science. So for starters, so I have questions for you. So it's not kind of scientific, but so recently you moved from university to company. So what really attracts you to take that position in company?
That's a great question. So I moved to a very particular kind of company that also matters, that I moved to a place where there's a lot of academic science going on. I think that matters too. But here's what motivated me. What happened to me over the last several years, maybe I'll start saying the following thing. I actually believe the things that I say. So I think when we say things, we write in papers, this would have a great impact on disease, or we say it in talks, or we write it in our grant applications as academics. I think sometimes people say that, they believe it, but they're not taking it very seriously upon themselves. They believe someone else is going to show that this has real implications for disease. I will just do my thing. And what happened to me in the last several years is that I realized that in the past, there was a real palpable and true gap between the things that we could do as basic research and the things that we could do in the context of human biology. Human biology was kind of at a disadvantage, and it was mostly because of measurement tools. So the types of measurements and manipulations that we could do in a model organism like yeast, we just couldn't do them in a human system. We couldn't do it with samples from human patients, and we surely couldn't do it with with models in the lab that were human models. And as a result of that, we could advance certain types of biological questions really far by choosing the model organism basic research path. And we developed in our minds this view that human biology is somehow less basic biology or less fundamental. It's kind of a more of a derivative. And I think for many years, that's the best that we could do. What happened, at least in my own research in the last several years, is that I realized that we're kind of at what we call an inflection point, right? When the exponent starts rising. And that we can actually study human biology directly with the same rigor and tools and depth that we have historically applied for more academic basic research. And once that occurs to you, then you ask yourself, why not just do it? Because when you think about it, studying human biology and human disease is interesting at two levels. The first is that we would bring real benefit directly to patients. There's something extraordinarily gratifying around this. People who go to medical schools, which I did not, I'm a Ph.D., not a medical doctor. People who go to medical schools, and work as clinicians, as physicians, they have this direct ability to impact individuals in an extremely beneficial way. But for researchers, it's often very many steps removed. And the idea that we could bring these things and bring benefits to patients, that was a little bit of a no-brainer. The second aspect of it is that there's something extraordinarily fascinating about studying humans, even if you just care about basic biology, because it's, these are complex organisms. They are, the genetic variation is natural. They are not controlled in the same way that we control models. And as a result, the system is a lot more complex. So even just from wanting a difficult challenge to solve a difficult problem, it's actually fascinating. And then together with it, I reached the realization that the time has come, that you can take these different pieces and put them together and possibly really transform the way that we develop new medicines, maybe something similar to the revolution that happened when molecular biology first showed up on the stage, and it became possible to take molecular tools and molecular explanations and think about biology in this way, but also develop medicines in this way. And I think the same is true now with these large-scale data sets, with these sophisticated computation, with the large new kinds of therapeutic modalities like cellular therapies. And it's kind of time to do a shift like that. And so in thinking about the things I've done and where their natural projection is going, this was a very nice extrapolation of taking the spirit of basic research to a place that deeply respects and does basic research every day, but that has this extra mission of delivering directly medicines for patients with the right level of patience and involvement and investment and so on. And that's what I ended up doing. I still have a lab, I still do research, but I want to see the things that I say the impact is. I really want to see that impact.
Thank you.
Okay, so Professor Okano.
Okay. Hello, this is Hideyuki Okano. So congratulations. So actually, I'm leading the brain project in Japan. Okay, so together with Atsushi Miyawaki. Actually, as you know, so in Japan, brain project we are using non-human primate and also genetic method. We recently developed the Autism Model and Alzheimer's disease, Parkinson's disease model. And so starting the single cell analysis, we found that. So based on the single cell analysis, we found a particular type of subset of cells are very important for the phenotype productions. But we still don't know where they are distributed and so make synaptic connection. In that sense, so the future, the combination of the imaging technique and so single cell technique or in situ hybridization will be very important. So as we noticed, there's lots of effort in combination of the in situ method and so single cell analysis such as Carl-Diceros STARS method. And do you have any thought how the distribution of the gene expression and how it corresponds to the neuronal circuit or in vivo situation? Do you have any thought, a new idea how to connect that?
So I'll share a little bit about work that we do in this area. The first is that I think we early on saw that the single cell profiles actually carry a lot of information about their spatial location. And if you give it a little bit of auxiliary information, for example, direct in situ measurements, but not necessarily for that many genes, you can start placing things into space. And in work that is not actually from my group, but from a good colleague, Mor Nitzan, they even showed that if you can make some simple assumptions, for example, of continuity, then without any in situ data, you can start reconstructing the ordering of cells. But you have to make those assumptions, which in the brain sometimes works incredibly well because of gradients and sometimes is not helpful at all because of, salt and pepper patterns. So it's not that simple. We've developed years ago in 2015, we actually developed the first method that took a limited amount of in situ data and cellular profiles and used the shared genes, the shared variables in order to port both the cells into their location and then bring along with the cell the full molecular profile. We have recently developed a much more enhanced algorithm for this that works pretty much with any in situ method that's out there that does that. We call it Tangram. It's a famous game, Tangram, where you have the puzzle pieces and you try to put them into place. So you think in the same way. You have the cells and they're like puzzle pieces, and you at first put them randomly into space. And then you use the information from the in situ in order to try and shuffle the puzzle pieces around so that they fit well with the in situ information. And that's based on more modern machine learning algorithms. That seems to work extremely well. And the cool thing that we can do on top of it is that we can add to it ways that then map it to histological and anatomical information. Because when you collect the in situ data in many of the methods, you also collect histology information. You can stain the section, for example, at the end. And now you can take this stained section and automatically, with another machine learning algorithm, map it to sections, say, in the Allen Brain Atlas. So you can tell not just this is my section and this is my in situ and this is where my cells sit, but you can say, oh, and this is where it is in the anatomy, you know, in, say, Bregman coordinates. And then you can say, and this is what I know already about all of these regions from, you know, decades and decades of neuroscience and so on. So I think that direction is going to advance very rapidly. And what is coming after it, actually, which is quite exciting, is that once you do this mapping, you can learn things from these maps, right? The cell characteristics are not the same when you define it just based on the properties of the cells intrinsically, the expression profile, the morphology. And when you also say, oh, tell me something about your neighbors, and I'll tell you more about yourself. And so some properties are going to be the same for a particular type of cell, almost no matter where it is. But as we know from the brain, the circuits matter just as much as the neurons, the individual neurons do. And so knowing something about the location and about the connectivity can allow you to redefine the identities of the cells. And this is where the methods are moving as the next step. And we have the beginnings, we have the first algorithms that have to actually do things like that. And they can now, I can now say, well, if I see this cell here, I can predict the state of the neighboring cell, or if the cell within a radius of 15 cells from it, and so on. And then the last point that I'll make, because you mentioned modeling autism, is that we've started doing perturbation experiments in vivo in animal models, so that we deliver a whole cell of variants into, say, a mouse, and then we recover back the tissue. And we've done this actually in the brain with autism genes that have large effects from loss of function, you know, that de novo genes with loss of, which are basically loss of function. And when you recover them back, it allows you to tell which cells were actually impacted by the perturbation. And you can do it not just one gene at a time, which is the historical way in which we had to, say, build a mouse model for each perturbation. You can actually deliver a lot of them at once. And where we're headed with that next is, after profiling individual cells, we're hoping to move back to imaging and actually to be able to section those. And going, you know, to Dr. Miyawaki's approaches, to section those and have imaging information available for the same sections, again, so that we can see the perturbation in the in situ context and the way that it affects things like the morphology of the tissue, the function of the tissue, and so on. You know, these remarkable things that we can do in animal models.
Okay, thank you.
So, any question, comment from Dr. Honda or Dr. Kubota?
Yes.
Okay, please.
Hi. My name is Keio Honda from Microbiology and Immunology Department. So I'm a microbiota guy. So I've worked with Lamnik Zabel, one of your colleagues, and also Elan Elina. They are long-term collaborator of mine. So, yeah. And I would like to ask you about your perspective related to the microbiota field. So people usually conduct abundance-based association-wide microbiome analysis. But those association-based approach lead to just a correlative species. So therefore, precluding the translation to the therapeutics development. So, and therefore we stick to the very much classical method, like notobiotic approach based on germ-free mice and culture bacteria. Those are burdensome and sometimes disgusting, the food. And but we stick to these classical method and to find out the causal bacteria that can affect the disease or some homeostasis maintenance. So, do you have any idea what's the next step in this field? And yeah, I'd like to know your thoughts.
So I have several thoughts. The first, which is a technological advance that we have actually started developing, and Sanja Vichkovic, who still is a fellow at the Broad, really led, is to try and get to spatial measurement methods that would allow you to look in situ at both the microbiota and the host cells. So you can imagine, since you mentioned Ramnik Xavier, you can imagine in Iran, you can imagine taking, say, a resection or a biopsy from a patient and being able to section it and seeing both the microbes, say at least in terms of their categories from 16S, and the host cells simultaneously. This does not give you causality, but it is a much more stronger form of data for making inferences than when all you have is the bulk measurements and you cannot actually make the associations. That, I think, as a lab technique is going to be available. It's in pretty advanced stages and would be quite useful, I think, for at least some subset of problems. The second point that you raised, and I think is the most critical one, is of course that for causality, we need to go beyond that. We have to get to genetics. And the question is, what is our path to that? And I think there's two sides to this coin. There's intervention from the microbiome side, and there's also intervention from the host side. So intervention from the microbiome side involves either being able to engineer the bacteria or being able to engineer the bacterial community or doing any chemical biology style of intervention applied on the microbial side. And I think all of these are being done at the moment. So what would I have to contribute? But as we understand better, at least the potential molecular connections from the host to the microbiome, the many secreted peptides that are going both ways, all these volatile compounds that everyone is making for everyone else, we have the possibility of intervening also on the host genetic side. Again, for example, through these kinds of pool perturbations and measuring the impact on the microbiome. When we do it in animals, we have to do it with, we do it with our own interventions, with engineered interventions. But I would imagine, and I believe that there is the movement in this direction of really combining human genetics together with microbiome research. And so you need a much larger number of individuals, of course, to make these kinds of genetic associations, but you have the ability of looking at the host genetics as your main source of perturbation, many perturbations at once, and look at the microbiome on the other side. A weak version of this, which we're seeing good success in for single cell analysis, and one would hope would generalize to the microbiota, is simply by looking at a large enough number of samples. Of course, people have done that for microbiome research. People are beginning to do it for single cell genomics research and spatial research, where you just have a large enough number of subjects now, but you could imagine putting them together. And so what we see from just at the host side is that you can find patterns of variation between different cells of different kinds in the host. When these cells activate this program, those cells repress that other program. If you also have microbiome information, then you can add the changes that happen in the microbiome, and at least those would direct the next generation of perturbation experiments. So these are some of the things that I could imagine would begin emerging in the field. Maybe the field also of molecular devices can improve to bring in some engineered communities in controlled ways. But I think it's a really tough one. It's a really tough one. It's not an easy one to really go beyond where we are today.
So nice to meet you. So I'm Yoshiaki Kubota, committee member of this prize. I'm very happy to be here, and I'm very happy to have you as a winners of this prize. So can I ask a non-science casual question?
Of course.
So one big problem to do single-cell RNA-seq in this country is it's really expensive. So it costs around $10,000 for one sample. So I'm not sure what's going on, but very limited numbers of laboratories can do that. So do you have any good idea?
So it shouldn't cost $10,000 for one sample. You might be doing too much per sample. Let me start with that, because it used to be very expensive, and it actually dropped many, many orders of magnitude. But of course, what happened as the orders of magnitude of cost decreased, the appetite grew. So what happens today is that you drop the cost of per cell by 2 orders of magnitude. People want to do 2 orders of magnitude more cells, and then they want to do 4 orders of magnitude more cells. That's an interesting commentary on humans, I think, more than on human cells. Having said that, actually there are 2 drivers for the cost of these kinds of experiments. One, in which I think my work has little impact, is the cost of sequencing itself. But my comment on that, which does come from my work, is that most people sequence too much depth per cell, and you can sequence a lot less, and you would still get the same outcome. Single-cell RNA-seq in particular is a method based on very sparse molecular biology sampling, and it's successful because of the structure in gene regulation, because genes go up and down together. And that's actually a statistical fact. And because of that, you can actually go for far less depth per cell and save a lot of yens or dollars, and still get basically the same set of answers that you wanted to begin with. That's one of these wonderful, magical mathematical things. It's not magic, it's actually math. The second piece is the cost of actually doing the single cell experiment itself before the sequencing. And there, I would say that the methods are only getting better and better and cheaper, and there's 2 ways in which we're seeing benefit. The first way is in what we call overloading, is the fact that you can actually use the same costly lab reagents to eke out more cells, and there's different types of tricks of how to get there, and they're getting better and better. For example, you can barcode the cells with one barcode, which is not unique in the first run, and then you can overload them, and those barcodes would allow you to actually distinguish those cases where you actually mix 2 cells together and you would still get a great benefit. We've shown that when cells are incredibly rare, sometimes you just want a very rare population, but you have no way of enriching it for it. We did it with enteric neurons. You can just do it like so and very easily find the ones that interest you, the needle in the haystack, without paying a lot of money for the haystack. So there's a range of techniques like that that basically go and use the same amount of lab reagents, but just more efficiently. You can carry that to extremes, again, with the help of math, and kind of do the not single cell experiment, where actually you're measuring little groupings of cells randomly, and you can still get their individual profiles. So that would be one dimension. The second one is that there's lab methods now that are actually cheaper. So the first massively parallel method was DropSeq, and it relied on droplets, and droplets became a very popular technology, and a lot of labs use them. But combinatorial indexing, which is a later technology, can actually be executed more cheaply. So when you have a very large number of cells that you want to study in one sample, then these kinds of techniques are sometimes an easier path downstream, and different labs have innovated along these lines in different ways, but that allows you to execute much cheaper experiments very efficiently at a cost that can be an order or 2 orders of magnitude less per cell. So you shouldn't pay $10,000 per sample. That is the conclusion. Not today. It shouldn't be that expensive today. Sequencing costs, that's a different question. One would hope that sequencing technologies will advance as well, but that is the area of other people, not me.
Okay, thank you very much. Yeah,
Yeah, thank you very much. So yeah, actually we can feel a lot of energy and also enthusiasm from you, and driving your cutting edge science. And also I like your word when I asked what's your great research ambition. You said you want to see I want to see biology becoming more predictable. Mm-hmm. That is very challenging, but I think where there's a will, there's a way. So yeah, you should be the one who is gonna do the cutting edge research for this direction. So lastly, I'd like to ask you, Professor Miyawaki, one question. So which aspect of science, your field in general, do you wish the general public know more about?
Well, so I I respect nature, because we are going to make the best use of any growing animals, plants, fungi, protists, and the bacteria too. And so we respect nature, and I like the general public, so know more about the natural resources on our planet. We need to do environment preservation in the future. So that's my my message to the public. Yeah.
Yeah, thank you. So we learn a lot from the nature and still. Okay, so thank you very much for both of you, and I think that's the end of the free talk. I think we should we wish we have more time, but I'm sorry, it's a limit of time. So thank you again for joining us today, and again, congratulations on the award. Thank you very much.
Thank you so much.